Greeting to Dr. Suresh. My name is Rmsd. Today we will be presenting our molecular docking project on the proteins and their associated ligands along. Next, starting off with the introductions, docking in short is studying how ligands and the protein interact and fit together. There are three types of docking, namely protein ligand, protein-protein, and protein nucleic acid. Next, the objective of our project is to illustrate the flow of the molecular docking software and show our protein ligand conformation that we have successfully docked, as well as computing various parameters relating to docking. This slide shows the databases that we have browsed to search for our protein and ligand. Following that, this slide shows the various software that we mainly use to compute our docking and analysis. Moving on, we'll be presenting the methodologies of our project. Firstly, in Discovery Studio Visualizer, we have set up active site coordinates as a grid box to allow for docking later. Proceeding forward, we started with the protein preparations by checking and preparing the missing atoms present in the chosen proteins in Autodock. Next, we will remove the ligands associated with the PDB format file. Okay. Then the polar hydrogens are added and the subsequent additional of the charges, namely Coleman charge and Jastiger charge. This marks the end of the protein preparations. Likewise, the ligand is prepared in the Autodock and the torsion count is checked to ensure it has a rotatable bond of at least one for docking. Next, I will pass to the next presenter, Wei Long.
Good session. I'm Wei Long. After ligand preparation, autogrid preparation is done by setting the specific target site coordinates that we retrieve from the grid box done in Discovery Studio. Next, autogrid is run. Following that, autodock preparation is done afterwards by setting up certain parameters such as genetic algorithm, ST search parameter, docking parameters, and the Lamarckian genetic algorithm as the output. If the parameters were set up correctly, the results, such as the one shown on the slide, can be viewed. Subsequently, Autodock is launched and a brief moment is required for computing the ligand-protein interaction. The conformer with the lowest Rmsd and lowest binding energy is taken into account. Following that, the conformer is then built in Discovery Studio or Autodock to visualize the docking result. The protein-ligand interaction can be viewed in 2D or 3D conformation using Discovery Studio Visualizer. In addition to that, various interactions such as hydrogen bonds, aromatic and hydrophobic bonds may be viewed as well. Finally, Rmsd value is calculated via PyMol with align or fit command to ensure that the value is less than 2. We will now proceed to the first docking, which we perform using the heat shock protein HSP9D that classifies as a chaperone protein and was expressed in E. coli. This protein is involved in cellular processes such as cell cycle control, cell survival, hormone, and other signaling pathways. It plays a key role in the cell's response to stress and is the main player in cellular homeostasis maintenance. It has become a major therapeutic target for cancer and an increasing interest for neurodegenerative disorders. HSP9D works as a dimer and has essential ATPase activity. Meanwhile, the ligand that we chose to target is a benzoquinone anti-neoplastic antibiotic isolated from Streptomyces hygroscopius. The drug binds to and inhibits the chaperone functions of HSP9D. Since HSP9D maintains the stability and functional shape of oncogenic signaling proteins, its inhibition induces proteasomal degradation of oncogenic signaling proteins that are overexpressed or active in tumor cells. So that's a wrap for my part, and I'll pass it to Stanley.
Thank you, Leon and Stanley. Using Autodock to carry out then run, it turns the conformer exceeded the lowest RMSD value, which is 0.00, and was taken as a reference conformer. In addition, we also run the PyMol to calculate the RMSD value of complex 1. Fortunately, the PyMol result determines the RMSD value of complex 1 is less than 2, which is 0.880. Next, after that, the complex 1 was viewed as a 2D structure using the Discovery Studio. This was done to analyze the bond between the complex 1 and the residue in HSP9D. A conventional hydrogen bond was formed with the 131st residue of the amino acid chain, which is the alanine, unfavorable acceptor to acceptor bond, and unfavorable pump happens to a common occurrence between complex one and the rest of protein. Besides that, protein like pi alkyl and carbon hydrogen bonds are also present between the complex and the protein. Next, the second docking that we have done is between the phospholipase A2 with the resveratrol. The phospholipase A2 play a role in phospholipid cleavage and will lead to the synthesis of icosanoid. However, if the icosanoids are synthesized effectively, inflammation will occur. The drug ligand that we have used to target and inhibit the protein is resveratrol, which is an antioxidant usually found in berries. Using AutoDoc, we have managed to compute the protein ligand interaction after a series of preparations that mentioned earlier. Out of the interaction, complex 32 has the lowest binding energy and lowest Rmsd after 50 runs in AutoDoc. The drug resveratrol acts as a hydrogen donor to the protein, whereas the protein acts as a hydrogen acceptor. The binding which include the interaction comprise conventional hydrogen bond, sulfur, pi sigma, and pi alkyl. And this show the 3D diagram of the result of docking, and the Rmsd derived is 0.417, which show that the resveratrol is a valid drug candidate. The third docking that we have done is between the pyrine and the ethanidiol. The pyrine usually acts as a signaling protein for the mediating intracellular signaling that has been linked to cellular response, to pro-inflammatory signal, and immune or stress response. It is chosen as a protein for molecular docking as it has a resolution of 1.56 angstrom, which is more stable for autodocking. Plus, the attraction method is X-ray diffraction, which is more reliable. The ligand we choose is 1,2-ethanidiol, also known as ethylene glycol. The ethylene glycol is a synthetic liquid substance that absorbs water and is odorless but has a sweet taste. The ethylene glycol is used to make antifreeze and de-itching solution for car, airplanes, and boats. During autodocking, the runs are then studied to understand the approximate binding energy. Run number 20 has the lowest binding energy, which is at negative 2.74 out of all 50 runs organized. The results are validated with the PIMO, where the Rmsd value of the ligand protein structure is 0.184, which is lower than the threshold of Rmsd, which equal to 2, indicating successful protein ligand docking. After autodocking, the structures are further analyzed in Discovery Studio to further comprehend the ligand protein structure. Firstly, the structure is analyzed in 2D form, where we can observe the binding of amino acid with the ligand. In this case, the bond of amino acid and ligand is in the form of conventional hydrogen bond. As you can see, there are also indications of the length of the hydrogen bond to prove the distance of bond between ligand and amino acid. That's all from me. Then I'll put the session.
Okay, thank you, Sandy. The structure is also analyzed in 3D form. In this picture, you can clearly see the interaction of the conventional hydrogen bond between the amino acid and the ligand. The pink colored surface on the protein states that the amino acid is the hydrogen donor in the bond, while the green surface of protein shows that the amino acid is the hydrogen acceptor. Next, the last docking is between 4-hydroxy-mandelic oxidase and flavin mononucleotide, FMN. This protein is classified as flavoprotein, and the resolution of this protein is 1.39 angstrom. The protein is extracted via X-ray diffraction. Now I'm going to briefly explain about the fourth protein we chose. So 4-hydroxy-mandelic oxidase catalyzes the oxidation of p-hydroxymandelate in the vancomycin biosynthesis, which is part of the antibiotic biosynthesis. Vancomycin is classified as glycopeptide antibiotics used to treat colitis, which is the chronic digestive system disease caused by inflammation of the inner lining of the colon that could occur after antibiotic treatment. Next, there is only one ligand that exists in this protein, which is the flavin mononucleotide, FMN. It also only has chain D, and it acts as a cofactor that helps certain oxidoreductases, such as NADH dehydrogenase, in oxidoreduction. It can usually be found in tissues such as muscles and cells, for example, in red blood cells and platelets. Okay, from this slide, the yellow ball in the middle of the picture is the sphere. The table shown is the sphere object attributes. The most important part is the XYZ coordinates, which indicates the direction of the sphere size. Moving on, the grid box is generated by adjusting the XYZ coordinates to the value that we received and observed from the sphere object attributes table. The number of points of the XYZ dimensions can be adjusted as well, as long as all the three values remain the same. So from this slide, it shows the Rmsd table and the clustering histogram. There are 10 conformation clusters created, and there are 2 clusters in this case. From the table, we can note that the first one is the preferred structure, which is the 7 runs, since it has the lowest binding energy and the least Rmsd value. This is the preferred structure in more detail. The estimated free energy of the binding is positive 345.75 kilocalories per mole, which is the lowest among the 10 conformations. The picture on the left shows the protein conformation, while the one on the right shows the ligand. Flavin mononucleotide acts as a hydrogen donor, while the protein acts as the hydrogen receiver. So this slide shows the ligand in an aromatic structure. The middle picture shows the receptor-ligand interactions in the 2D diagram. The interactions include an unfavorable bump, which is in red color, conventional hydrogen bond, which is in green color, then the light blue carbon-hydrogen bond, pink pi-pi stacked, and alkyl and pi-alkyl, both in the light pink color. The last picture shows the ligand itself. So molecular docking has several limitations that need to be addressed. Firstly, the lack of a quality database. There are several cases of false positives detected during the autodocking due to highly sophisticated ligand drugs used. This reduces the accuracy of ligand protein docking, thus making it incapable to be used for further studies. The next problem is related to the lack of accurate scoring function. When PyMol is used, the Rmsd scoring of PyMol sometimes varies, and this indicates that during practical use of a large number of binding poses are evaluated, there will be inaccuracy in binding energy and stability scoring of the protein ligand molecules. Lastly is in the challenges regarding model interpretation. So identification of the target protein and the active site with an ideal ligand is not relevant enough for drug discovery, as the protein fluctuates through autodock depending on the relative free energies of each of these states, spending more time in conformations of lower free energy. So in conclusion, we have learned that molecular docking-based technologies have become the direct method to efficiently discover potential drug targets. We have successfully bind ligand to protein and have extensive learning of software users to help in mastering molecular docking. So that is all from our group, and thank you for listening.